Good morning and welcome, my dear medical students. The chikungunya, a self-remitting febrile illness, having its origin from a virus of alpha virus type of the family Togaviridae. So, chikungunya self-remitting febrile illness, self-remitting febrile illness. The word chikungunya is derived from an African dialect, Swahili or Makonde. So the word is derived from the Swahili or Makonde. It is a dialect of Tanzania in Africa. The meaning of chikungunya is to be bent over. So the meaning is to be bent over. The other name for chikungunya in Congo is buka. The other name for chikungunya in Congo is buka. The translation of buka is broken. The meaning of all these terms, chikungunya, buka, and broken, to be bent over, is stooped our posture. This stooped over posture is exhibited by the individual of this disease as a consequence of severe chronic incapacitating arthralgia, severe chronic incapacitating arthralgia, so the stooped over posture is exhibited by the individual who is suffering from the chikungunya, and this is a consequence of severe chronic incapacitating arthralgia. The roots of the chikungunya back in 1953. One, it was first discovered in the Swahili village of Tanzania. The etiology of the chikungunya, it is a viral disease caused by alpha virus of Togaviridae family. So chikungunya, chick V and chick F. So what is ChikV and what is ChikF? The name of virus and name of the disease or illness caused by this virus is chikungunya. So when I say ChikV, I am talking about the chikungunya virus. When I say ChikF, I am talking about chikungunya fever. So both the name of virus and name of the illness caused by this virus are referred as the ChikV and ChikF. So it is an alpha virus, or I can say alpha virus. The family of alpha virus is Togaviridae. It is an arbovirus, that is arthropod-borne virus. Arbovirus. That is arthropod-borne. The man is the main reservoir for the virus, and it is transmitted from one person to the other person by the bite of day bite mosquito. The Aedes is day bite mosquito, or the bite of the Aedes is in early morning hours or maybe in early night hours, but mainly it is day bite mosquito, mainly in urban areas. It is transmitted by the Aedes aegypti. But in West Africa and in East Central South Africa, it is also transmitted by bite of some wild Aedes, like Aedes africanus, Aedes neo-africanus. The chikungunya virus is having 4 different groups, or it is divided in 3 groups on the basis of the partial sequence of the genetic material according to the sequence of NS4 and the E1 gene. So phylogenetically, the chikungunya virus is having 3 groups, and these are divided on the basis of the partial sequence of the NS4 and E1 genes in the virus. A single-stranded RNA virus having single-stranded RNA virus having a length of 11.5 B. And it is enveloped in the capsid and phospholipid. So it is having envelope of the capsid and phospholipid. So there are three groups of the chikungunya virus. The first is West African type. The second group of chikungunya virus is East Central South African type. East Central South African type. And the third one is the Asian type of the chikungunya virus. So ChikV is having three groups: West African type, East Central type, East Central South African type, and Asian types. These groups are divided according to the genetic sequence of the NS4 and E1 genes. The chikungunya virus is frequently involved in the outbreaks in the tropical and temperate countries all over the world. The virus chikungunya is involved in the frequent outbreaks in the tropical and temperate countries of the world, like the countries of the Africa and the countries of this Indian subcontinent and Southeast Asia. But in recent, there are some travel spread cases of the disease. So it has drawn the attention towards the European and American countries also. So it is involved in frequent outbreaks the entire outbreak or entire epidemic virus is maintained by some different cycles according to the territories. There are some different cycles which maintain the entire epidemic period of the virus. In Africa, it is maintained by the epizootic, where the virus is maintained by the monkeys, the rodents, and the birds. The monkeys, rodents, and the birds. The other cycle, which is also occurring in Africa to maintain the inter-epidemic virus, this is the sylvatic cycle. Sylvatic cycle. In sylvatic cycle, the virus is maintained by the monkeys, rodents, birds, and non-human primates, or I can say the wild primates in the Africa and in the wild Aedes mosquitoes, like the Aedes taylori, Aedes africanus, Aedes neoafricanus, Aedes pelricum. But in Asian countries, and in Indian subcontinent, the virus is maintained by urban cycle. So in urban cycle, virus is maintained by Aedes aegypti mosquito, and human beings are the main reservoir of the Chikungunya virus. The entry of the virus in the epithelium after the bite of mosquito is unclear to date, but there is some recent research from the Indian subcontinent and Asian countries which have proposed the entry of the virus in the epithelium. This is the entry is clathrin independent. This is about the entry of virus in the epithelium. This is clathrin independent, but it is ESP15 and dynamine dependent. Dependent endocytosis. The entry of the virus, Chikungunya virus, in the human epithelial cell after bite of the mosquito is clathrin independent, but it is ESP15 and dynamine dependent endocytosis. The pathophysiology of the disease. The pathophysiology of the disease. To date, there is very little knowledge about the pathophysiology of the chikungunya and the other viruses, and it has to be investigated to date. But there are many research from the Indian subcontinent and some other Asian countries which has proposed some different models for the pathophysiology of the chikungunya virus infection, or I can say chikungunya fever. One of them is murine model. After evolution of the T cell and B cell mediated immunity, which has shed light on pathophysiology of the chikungunya virus infection and chikungunya fever. One of them is the murine model. According to this model, according to murine model, there are anti-chikungunya virus antibodies, anti-chikungunya chikungunya virus antibodies have been detected in the blood which are directed against this is the E2 glycoprotein of the virus, and it is having its C terminal. So this is the C terminal These anti-chikungunya virus antibodies are directed against the C terminal of the E2 glycoprotein of the chikungunya virus. So these are directed against the C terminal of the E2 glycoprotein of the virus. Both natural antibodies as well as the virus specific antibodies, that are the anti-chikungunya virus antibodies, are necessary for controlling the chikungunya virus disease or chikungunya fever. The other model which has been proposed to explain the pathophysiology of the chikungunya fever is the ex vivo multiplex study of the chikungunya fever. It is a large study, and it is a study of the T cells, 50 cytokines, chemokines, and growth factor plasma profile. So it is a study of T cells, 50 cytokines, chemokines, and growth factor plasma profile. So according to this study, this is the ex vivo multiplex study of the chikungunya fever based on T cells, 50 cytokines, chemokines, and growth factor plasma profile. According to this study, the chikungunya virus elicit strong innate immunity. Innate immunity is immediate type of immunity. So it elicit the strong innate immunity, and this immunity with abundant production of the pro-inflammatory chemicals or markers